So one very unfortunate thing is that P. acne bacteria doesn't necessarily stay exclusive to the skin. There are other areas that are associated with it and other diseases that are associated with it. They can affect the sinuses as well as after surgery. There's arthritis components and brain inflammation and infection. So there are a number of different areas that P. acne bacteria can affect. Specifically here, the one that's probably more associated to acne itself is a sinus infection. There are treatments such as interlesional corticosteroid injections. There's also cryosurgery and isotretinoin, but these are not always effective. One risk factor for them is smoking, another is obesity, also being black is a higher risk factor, is an increasing risk factor. Now, it occurs roughly in about 1 in every 600 people. This is something that's just, it's not necessarily important to know for your specific disease or condition, but it may help if you have something that seems exceptional, or if you have maybe a lot of sinus infections. It may be something to get looked at to see if maybe P. acne may be a source. Now, how that would alter your therapy, it's hard to say, but it may be that instead of an antibiotic, that a isotretinoin treatment may be a better source of solving that condition. So this graph takes a step back when reviewing the immune system. So you have something that a lot of people understand, which is stem cells. Stem cells are the holy grail. We've heard these things about from as a kid, that they're going to save the world because they can become anything. Well, every different cell line has a certain level of stemnicity, if you will. There are bone stem cells, and then there are brain and blood stem cells, but the reality is only certain kinds of cells can transform into virtually anything, or have that level of plasticity, having committed to their cell line as much. So we have stem cells, and these are probably coming from the bone marrow. Now, these will go into one of two different things, probably more, but for our purposes, this is what they'll do. There'll be a lymphoid stem cell, or there may be a myeloid progenitor cell. Now, from there, you get into things like lymphocytes, which we've all heard of, I'm sure, or many people have heard of a lymphocyte, something in, a cell that is in the lymph. And then you also have granular cytes. So we have beta progenitors, T cell progenitors, which can break into other cell lines, and also natural killer T cells, or natural killer cells, I should say. Again, you have plasma cells, you have memory cells, you have TH cells, you have C cells. We've probably heard of memory cells. These are cells that hold on to the immune system's memory, in the sense that if you get an infection, you become immune to that because you have developed those antibodies. Those antibodies are present within the memory cells. So if these memory cells are sitting around and they have an understanding of this same antigen that comes in, they'll start producing the antibodies rapidly. It's sort of like a reservoir of the same antibody. There are other elements to memorization of an antigen, but that is, memory cells play an important role in that. And so it would be important from a perspective in allergies to analyze memory cells as a component that may be able to be altered. Because if you have a memory of an antigen and you consume something and then you have a volatile reaction to it, it may be in the memory cells that you're having this reaction. So although it's a genetic component, maybe it is a genetic component within the memory cell. I'm not saying that specifically that's guaranteed, but it's something, an area of interest to look into. Now, you have TH2 and TH1 and TH2 cell lines as well, but this is kind of what they're saying here was TH cell and THC cell. Natural killer cells, we've talked about those a little bit before, how they go and basically grab on to other cells that are infected and break them down and eat them up. Phasocutize, I'm saying that wrong, but they eat the cell again. So then you also have neutrophils, eosinophils, basophils, mast cells. These are cells that are strongly associated with allergies as well because of their ability to produce histamine. Then you have monocytes, which are dendritic cells, and macrophages, which also eat bad bacteria, among other things, and basically they lyse them within their digestive sacs that they have within their digestive vesicles, I should say. Now, all these again play an important role, and they all have subtypes. We'll try and get into as much as we can here without trying to be too broad scope that we lose the perspective that it'll have on acne.
And this is what I mean by talking to each other. So this is basically this is one of the most important ones. This is a antigen presenting cell, which can be basically any cell, including your skin cells, also known as keratinocytes. Your major histoimmune complex, MHC, is basically a cell. It's from the genes, and this is basically how your body knows from a genetic standpoint how one cell is you and not somebody else. And when this interacts with other cells among other binding sites, you get a proper respective transition from one cell to another, whether that be T regulatory cells, TH2, THC, which can go into all certain different things. As you see, there's a slew of different chemicals, and it's a headache to try and wrap yourself around it. But this gives you some understanding of what we're dealing with when we're talking about why someone's allergic to something, or how does the immune system properly attack a bacterial infection, per se, and not just go bananas on it and not cause a whole lot of inflammation. How does it make sure that it's specific enough to not attack you, and what might we do in order to limit its the collateral damage generated by this response. So here you have an antigen binding to a antigen presenting cell. Typically that's something like a dendritic cell, but that can be something like a keratinocyte. And when that binding happens, it's going to send out chemicals. As those chemicals are sent out, it's going to attract immune cells like T cells, and when those are attached, you're going to get differentiation, because it's going to be able to talk to the cell and tell it what to do and how to do it, and then it's going to change appropriately, hopefully, and create the proper response. As you can see, that this is something that you thought just happened in the lymph node, but it's not necessarily just there. It can happen in other areas, but just the discussion is a little bit more crowded. Basically, I would say, in the lymph node is basically how they can how the immune systems can sort of talk in peace without the all the excitement that goes on in the body, whether that be in a part of the skin, in a part of the brain, in a part of the muscle. More stuff. Don't really need to know all this, but again, if you do, well, freaking good for you. But it would help give you some context in order to solve things like how maybe a tumor cell was recognized, or how fibroblasts work, which are basically cells that are very important for the development of scar tissue, which could be important for individuals who have a lot of facial scars as a result of acne. So this is the gut. This is very important in respect to allergy development. As I'm saying, I'm sorry to keep going back into allergy development, but sometimes people are very annoyed when they're like, well, why am I allergic to something? And so one place to look is the gut, because this is where there's a whole lot of immune activity here, and because of the fact that the gut is such a central or sensitive area of the body, and really one of the gatekeepers to the body's health because of how permeable it can be, it has a lot of immunological activity. I mean, you put something on the skin, although it's very important that the skin although it's very important for the skin to remain separated from the external environment, it has a lot of dead skin cells on it that aren't very active per se, and also other barriers. But in the gut, it's more sensitive, and because the gut is so moist, and because there has to be so much nutrient absorption that occurs there, and so that's why a lot of allergens that's potentially one of the reasons why allergens are typically said to develop within the gut. So this just shows you how the gut basically samples microbial organisms. Although this isn't actually how it's done anymore, they found out that these dendritic cells don't just leach out and sample bacteria. Other cells come and grab them and then present them to these dendritic cells. However, that's still these slides are still relevant into the context of the development of an allergy. So this is a good healthy response. It could be potentially a healthy response as well, but this is still this is as a result of a bad bacteria coming in and attacking the gut. And so we have our neutrophil activation, our macrophages, and all that exciting stuff. Okay, so I don't want to get crazy on this, but this is a way that isotretinoin or Accutane effectively regulates the immune system. So this is retinoic acid. This is how it is able to block certain development of cells and alter the direction of how a cell specifically develops. Here we're seeing some different kinds of antibody development, IgM, IgD, IgG here, IgA, which isotretinoin inhibits, which is unfortunate because it's very important for the development of mucus, and so that's one of the reasons why you can really dry out in the gut or have gut issues. Now, I won't get crazy on this, but this is basically just a quick introduction to some certain terms. Another one is Toll-like receptor, which is important for the innate immune system, which we'll talk about a little bit. So this goes into hypersensitivity. Why does our body respond so aggressively to certain allergies? And I'm not going to bore you anymore. So I think what's important to recognize is that this is some ways that some of the most prevalent ways that you may become hyperreactive to an antigen. Now, I should mention that IgE is basically immediate. So if you eat something and all of a sudden your throat swells up or something like that, that means that, or you start getting itchy, that means that it's an IgE related antibody. Now, if it takes like a couple days or so, that primarily means that it's IgG related, meaning that certain antigens result in certain different kinds of antibody responses. And one of the most obvious things to do is like, oh, a lot of people think, well, if my throat doesn't close up, then I'm not allergic to that peanut or to that almond or to milk. But in reality, it's not the case. Although that's an important indicator, that allergy can develop on your skin differently. So maybe, again, though, it doesn't matter, because even if you instantly start itching someplace, or sorry, I should say, even if you don't instantly start itching some area of the body, that doesn't mean that you're not allergic to that food if you consume it, because up to 3 days and possibly even longer later, you'll actually have the development of the related antigen response. So you may not have you may not develop a pimple because you ate an almond, but maybe several days later, all of a sudden you start breaking out like crazy, and you haven't eaten almonds in a while. Boom, that's how you can make that association. There's also ways to effectively identify an antigen or an allergy through something like a skin prick test, or there's other ways as well, but they're not 100% accurate, and really the only way is to eliminate a food and find out if your condition gets any better, unfortunately. But you should do it roughly over a 2-week period, and or maybe up to a month, and by simply avoiding that food as effectively as you can, you would be able to see where that puts your condition. So this is what I'm talking about, is this delayed allergy or delayed hypersensitivity. There's at least 4 different kinds of hypersensitivity reactions. The most important one, or most common one, I would say, that is difficult to recognize is the IgG mediated, which can take up to like 2 days to present itself, and generally peaks at 3 days. So the other important thing to remember, though, is at day 4, it's not like you're going to be asymptomatic. You're still going to be itching, but you may be less itchy. So you may think, oh, well, I didn't really react, or I didn't feel any better after 3 days of avoiding a food. Well, you could still be coming down off your prior exposure. So that's why you need to do it at least up to 3 weeks, so all of that antigen and all of that response can be reduced to a baseline level and potentially improved on dramatically. And because of also all the other components of acne development, you may not necessarily see the traumatic difference between not eating it for 3 days and eating it for 3 days. So that's why it's important to integrate multiple holistic, again, forms of treatment in order to rapidly identify and rapidly cure your condition, because the more spot on with your diet and your whole health system, the easier it is to find the factor that is causing you to have the condition. Meaning that if your sleep is right, if your diet is right, if your glycemic index is low, if you're not drinking milk, if you're not stressed out, and all those other components, then we can definitely that cancels out a lot of other factors. So it really helps us understand what could be the issue. So it really helps us diagnose by eliminating issues. So that's what I'm saying. So this is really important for allergy development, and in the context of understanding it. So what I'm telling you is that when you have an allergy, you have an antibody to a specific antigen in a food, per se. Or maybe it's some sort of chemical that you're reacting to, fragrance or something. And that antibody is being produced by B cells, and that B cell is knowing how to produce that because of the dendritic cell is presenting the antigen, right? Right. But how does the dendritic cell know which antibody or antigen to present? And that gets pretty complex. So basically what I'm saying is, how does it know what issue to address? And that goes into troll-like receptors, and that also goes into the major histocompatibility complex. Now there's a couple different pathways for this development. So there's the classic 1 pathway, and there's this CD1D pathway, and there's also this MHC class 2 pathway, and we can follow these within the relative context of how they're colored. So this is blue, so everything that's blue is in relevance to how the MH1 class pathway. Green is CD1 pathway, and yellow is this class 2 pathway. Now, this is published in Nature, a very prestigious journal. I also want to thank the authors here. So, as you can see, I'm not going to talk a whole lot about this, unfortunately. But a couple things that have been shown that to be very important is how those antigens that I'm talking about are just basically fragments of proteins. So what this really means is basically how's that dendritic cell or that antigen presenting cell break down the proteins and understand which of those proteins to present. And this is one of the ways that those proteins are broken down, and this is one of the ways that those proteins are taken up. So they come in this way, and so the protein comes in, it's recognized, and then it's shot out, and then it's broken down. It's cut up even more by other molecules or enzymes, and then it comes in to the endoplasmic reticulum and basically gets into these receptors, and then that works its way through and is understood by the DNA, and then comes back up here via the Golgi apparatus, and then it gets presented on top of the cell and tells other cells what to do. That leaves a lot to interpretation, but it's certainly enough information for most individuals to research for themselves what to do, and that's something I'll talk about later, as well as how to do your research. One of the most important things for allergy development from a genetic standpoint is the major histocompatibility complex. This is formed out of these 4 types here, and also here as well. Extend to the essential antibody coming in or getting in. So one of the best examples of allergies currently, because of how prolific it's advertised, is gluten. So, but the thing is that many different grains have gluten in them. So you're not necessarily allergic to gluten. You're allergic to wheat gluten. But wheat gluten is a protein. Now, going back to the biochemistry of an antigen, which I haven't really completely explained to you, is that the antigen works by recognizing certain amino acids, things that make up proteins. Now, so it's recognizing these amino acids. It's not necessarily recognizing the whole protein. It's not recognizing the protein as a whole. So you're not allergic to wheat gluten. You're allergic more specifically to the gluten gliadin mix. But you're not allergic to this gluten gliadin mix. You're allergic to the gliadin epitopes. But you're not allergic to just any gliadin epitope. You could be allergic to alpha, you could be allergic to beta, you could be allergic to gamma or omega, and even more. So there's these little tiny differences within these proteins that could cause you to react. And so that goes into the issue that conventional laboratory testing is only capable right now of using or selecting for the most common antigens or epitopes. But if they're testing for beta, but you're allergic to omega, you won't find that out because it's so specific that it's not interacting with this. It's not finding the omega, and this is the issue: is that we don't have a complete holistic recognition system for gluten or any other allergy. We're only using the limited specificity that we have for each individual in order to get the best result. So the only surefire way in order to find out if you're allergic to something is the 2-week challenge. So if you avoid it for 2 weeks and you're better, then chances are you're allergic to some aspect of that gluten. We can't necessarily confirm or deny it, but that seems to be the case if you're improving. It's very unfortunate that this is how advanced we are within immunology, and immunology really needs to take a new stride in recognizing how to get the appropriate diagnosis for individuals that we can actually trust. Because certain people could be walking around not thinking that they're allergic to something, and they actually are, because the laboratory testing isn't able to properly identify it. Now that we know that allergy responses occur predominantly because of proteins, how does a chemical create a reaction? Because a chemical doesn't necessarily have the epitopes that are needed to recognize, for the antibodies to recognize their existence and therefore react to them. Well, as it turns out, the body has this Hepton response, which basically allows them to make a respective protein that is incorporated around or in the general area of those chemicals, and then the body can have an immune reaction to them. So basically the body forms a protein to react to the chemical, which is a very interesting paradigm. I won't get into how these Hepton responses occur or are formed, but this is one slide that I was able to gather, but doesn't necessarily give you a great understanding of the occurrence of Hepton proteins. So this is how the body is responding to fragrances. Fragrances aren't proteins; they're chemicals. So whenever someone is responding to a fragrance, it could be occurring from the innate immune system, meaning a non-antibody specific response, or if it's very aggressive, it could be responding with this antibody response. So now another much more perfect example of this is actually poison ivy. There's an oil on poison ivy, as most of the population knows, and it's this oil that we're responding to. And when this oil gets on our skin, then we begin this Hepton response. So then your dendritic cell goes into your lymph node. Remember, it teaches the T regulatory cells, and it teaches all the other cells how to respond appropriately. And then that response goes back into the region. Now these are via the afferent lymph. This is the infferent lymph. This is basically coming in, coming out. That's all that is. And then what you would call effector, basically affecting, active, not necessarily teaching, more or less just creating a response. Those are your histamine releases and all of that associated issues. Now, interestingly, actually we'll save that for a little bit later. So here we have a nickel allergy. This is one of the most commonly encountered allergies that occurs in man, and it's actually the most common metal allergy as well. Now, with chronic exposure, you can get about 5 to 10% of the population responding to nickel. So potentially, if you have too much nickel in your diet, I don't know how much nickel is in your diet, but that could be an issue. But also, if you have metals such as belt buckles and buttons and such around your maybe your back, that could be what you're responding to. I don't know why you'd have nickel all over your face, but maybe it's a chin strap or something, and that has some metal in it, and that could have nickel, and you could be reacting to that. So, now, you could also, because this could also be in sunscreen, nickel, so you could also be responding to your sunscreen with this reaction. So you have to be careful that you might need to get nickel-free sunscreen. I'm not saying that everyone needs to do that. I'm just saying that the prevalence is about 5 to 10%, and if you feel like every time you put sunscreen on you break out like crazy, and you've tried to a couple different sunscreens, and even the non-comedogenic maybe one, then look at this as maybe your target for how to prevent yourself from breaking out with sunscreen, and maybe that's what all of the sunscreens have in common. It's just something to look at. So let's go into the mechanism. So there's no epitope, so there's no antibody response. What there could be is a troll-like receptor, endothelial-mediated response. This is basically the cells themselves, not the necessarily what you would typically call the immune cells, but the cells that are in this specific region are actually reacting to, recognizing the zinc, or the nickel, I should say, and reacting to it. Why this is the case? Well, it's difficult to say and gets into a whole mess of interesting chemical reactions, but the baseline is the accumulation of effector cells in a target organ can determine the symptoms of zinc allergy. So basically, it's the level of these kinds of cells, and it's the amount of immune associated chemicals that is determining the prevalence of the reaction, but that is also determined by how many troll-like receptors are on the endothelial cells. So you can see there's this annoying cycle where what the hell do I target? Where's my therapeutic? How can I not have this allergy? That really probably comes to trying to reduce troll-like receptors, but in the short term, it could be more about reducing things like IL-10 secretion from the lymphocytes, and by that you could potentially reduce the total immune trafficking. I mean, how many immune sites actually go to the area finally to fight the reaction after being told to by the troll-like receptors that are on the keratinocytes, your skin cells. And so that could be the reason, or the way you could go about this is you could try and get rid of troll-like receptors long term of their recognition, which is pretty difficult because that's really genetically predisposed more than anything, but also the level of exposure and all that. And then, but the other shorter term is that you try and reduce the inflammatory chemicals by some anti-inflammatory agents, or maybe reducing histamine degranulation, and that could be your target. Not going to be perfect. It's not going to be at the root cause, but it's still going to give you some relief. Now, the most obvious relief that you're going to get is to avoid the damn zinc, or nickel, I should say. And by avoiding the nickel, you're then avoiding the immunological response. But that's not what everyone wants to hear. They want to hear, well, make me like everybody else. And as you can see, it's pretty difficult to do that. So now we're at the complement pathways. So there's 3 different complement pathways. And they can go from an antibody response to basically what's called the lectin pathway, or this basically is it's recognizing surface proteins. And then there's also the alternative pathway, which also recognizes proteins on the pathogen surface. So I'm not going to get into exactly how this works. Dr. Najib has done an amazing job at illustrating how this works with his proteins. But there's different ways for this formation to occur. But the interesting thing is when this is a several step process, and basically you always have a combination of there's a CA and there's a CB, and those numbers vary, as you can see, C5, C6, C7, C8. But as these CAs and CBs that are connected to each other break off, you basically have some which form a protein structure which works to basically making a complete whole, or I would probably call it a more a drill that goes into the bacteria and basically destroys them. Now, as this quote-unquote drill is forming, you're getting, with each of these breaks, that other part, because there's 2A and 2B, that B part is forming the drill, where the A part is more or less forming or asking for more inflammation and for more irritation. One of the things that has been studied is this increase in C5b formation in acne. And so this gives you a strong indication that there's a very high immunological response to bacteria because of this constant drill formation. The other very unfortunate thing about this, though, is that this drill acts on both our skin cells and the bacteria. So when this drill keeps, all these drills keep forming, you get more and more destruction of the area. How the body has corrected this is that is via blood cells. Blood cells can actually render these drills inert. So these drills basically get destroyed by blood cells. Now, so you're thinking you want more blood in the area, but remember that blood comes with inflammatory components as well. So it's a hard bit to really figure out how we can use the complement pathways as a therapeutic target. Now, a generalized one is get rid of the bacteria that it's responding to in order to get the complement system from responding to the bacteria. And that's one of the reasons why we use benzoyl peroxide, is to try and reduce this complement system and this formation of these tiny drill proteins.